Alrighty, everybody, I think it's 11 o'clock, so we're going to go ahead and get started. The Center for Integrative Environmental Health Sciences and Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar. Today, I'll be introducing our speaker, Dr. Jala, who serves as an associate professor in the Department of Microbiology and Immunology. His research areas include investigating the role and mechanisms of gut microbiota and their naturally occurring metabolites in regulation of human pathophysiology, with a special focus on exploring how dietary-dependent natural products and their metabolism regulate gastrointestinal and immune function. He comes to us with 74 peer-reviewed scientific articles that have been published, many different NIH-funded grants, as well as intramural and industry grants. He's served in several administrative committees, NIH and DoD study sections, as well as an editorial member and peer reviewer for several different scientific journals, and has a long list of awards and accomplishments throughout his career. And I believe that his work really speaks for itself. So with that, I welcome Dr. Jala to present our seminar series today.
Thank you. Thank you for the kind introduction. And then, yeah, things go pretty good. So today, I know I'm been talking about this arsenic and gut toxicity for quite a bit with this group, but whoever is not present, I'll just give a brief introduction about the arsenic and their role in the gut barrier dysfunctions, and then how we are kind of combating the activities with our newly identified microbial metabolite. Arsenic is naturally present in high levels in groundwater, and it is contaminated water used for drinking, food preparation, and threat a huge public health. And the risk of poisoning the worldwide is almost 230 million people. In Asia, it is 180, and in US, we still see it almost 2.1 million people are got affected with this arsenic contamination. So arsenic is not just new one; it's very old, and it's been used as arsenic poisoning. And also, actually, it is interesting that when I was looking at the old literature, I found that some of the things that arsenic was used to really kind of in the makeups in the women's in 1989 itself, there's a newspaper there. You can see the advertisements for the arsenic complexion wafers, and so then people started realizing how bad it is, and then they started saying that old witch, and it's a don't drink arsenic kind of thing. It's an extremely dangerous. So now, arsenic, if you look at the arsenic, it affects the numerous parts of our body, including skin damage, and then pigment changes and nerve damage, circulatory problems in the skin, and the cancer risks that have been reported with the lung, bladder, kidney, as well as the liver cancers, and especially with the Dr. State's group looking at, and Mike looking at the skin cancers. It's basically, it's not just a single, but not much literature is known about gut related issues. So that's where my lab started looking at it. There are the chronic arsenic toxicity can cause numerous diseases, as I mentioned here, including the cardiovascular, cancer, nervous system, and also whereas acute arsenic toxicity can cause nausea, abdominal stomach pain, diarrhea, and the list goes on. And especially the severe pictures, we can see how arsenic can cause skin damage. It's pretty nasty, and it's affected worldwide. It's not just in restricted to one country. You can see that there's a red indicates the high exposure level of risks that people have been in US. There's a significant high exposure there, and then increased risk. It's just, as I mentioned, it is a global issue, and in US also, as I mentioned, it's the 50 micrograms liter, which is considered as a toxic level arsenic, you can see that there are several spots within US we see arsenic contamination. So as I mentioned, the multi-organ damage, it happens. Especially my focus is to look at the gut area. So how the arsenic is impacting the GI tract and how it is causing other GI related problems. So my lab works on the gut microbiota, and it's been known and still discovering its role in several disorders that starts from AIDS to the metabolic disorders, Alzheimer's, cancers, and everything. And as I mentioned, arsenic can also cause uh, in a multi-organ damage, and I suspected microbiota would be also play a significant role in the toxicity or protection against arsenic-induced uh, damages. So I mean, our paper, one of the paper that people have shown that the role of microbiota in arsenic by Dr. Seth TWOG group, where they have shown that when they use mm, these are the arsenic mice, if you look at that, this is that when I use the antibiotics, you can see that arsenic is significantly reduced in the stool compared to the sham control. That means you can, when the microbiota is depleted, you can see accumulation of arsenic in the tissues. That kind of the first clue came that microbiota is required for the full protection against arsenic-induced toxicity in the mouse models. And that's basically, is required for metabolism and excretions. And they also looked at one of the mice that basically arsenic is metabolized by the enzyme called AST3MT arsenic SAM amethyltransferase, which is known to metabolize arsenic and responsible for the detoxification of the arsenic. So in this model, when they looked at the arsenic knockouts compared to the sham, when they depleted the microbiota, these mice are much more susceptible for the arsenic induced toxicity. I think it goes pretty even 25 ppm to 100 ppm, and you can see that as the dose increases, the survival of the mice significantly decreased. And also, when they did the human transfer, the human microbiota transfer into these mice, the survival significantly increased. So basically, supplementation of microbiota protected from mice from arsenic-induced toxicity. So in conclusion, the microbiota is required for the arsenic induced again, it works against arsenic-induced toxicity. So my interest came up thanks to CIEHS pilot program, where when they announced, okay, they're going to give some environmental thing. So then my expertise to look at the GI functions. So when I looked at the literature, what arsenic does to the GI, there's very, very, very limited literature that was available with respect to arsenic and gut functions. The one of the major thing that really attracted my eye is to look at the one of the Northport village, Northport Project, which is in the Washington state, which is a border between the United States and Canada, where there was a company called Tech Metals. They kind of significantly they smelter hose into over the Columbia River, and which almost produced 150 almost 145,000 tons of slag went into the Columbia River for an annual basis, but it has a significant level. Four independent studies conclude arsenic level is almost 100 times higher than the safety standards, and the cadmium is almost 18 times higher than the safety standards. And then when there was a big lawsuit going on, or it's when it was still, I think I'm not sure yet the status, but 17 people out of 120 people confirmed that they were have IBD diseases, which is almost 10 to 15 times higher than the national average. So that kind of suggests that maybe there is a link between the arsenic and IBD context. So that means it has a role. It might be causing, because unfortunately this IBD or any kind of these GI related disorders, it won't show up immediately. It is a chronic exposure and slowly it builds up and then suddenly shows up, and the root cause for IBD is impossible to track it back. So the major premise for the why arsenic could have gut function came from the literature, where I started looking at chronic arsenic expert drinking water exhibit symptoms of dyspepsia, gastroenteritis, and chronic diarrhea. And as I mentioned, gut microbiota is required for full protection against acute arsenic toxicity in mouse models. And subchronic exposure led to disruption of the colonic epithelial structure and barrier function. And there's a trivalent arsenic form significantly induce inflammatory cytokines such as IL-6, IL-8, and which I think I will discuss about this data which we generated. And arsenic metabolites, and they do target GI tract, leading to the enlargement of intestinal wall, edemas, and hemorrhages, and necrosis, and ulcerations. So the role, how, I mean, yeah, yes, gut microbiota is required, but is the bacteria is required, or is there any kind of microbial metabolites that can we combat with metabolites or not, is the question that I'm trying to answer in my studies. So our testing hypothesis is as follows, where you have exposed to the significant metals, they can cause oxidative stress and altered microbiota, leading to the inflammation, and which could be responsible for the leaky gut, and which leaky gut leads to the increased inflammation and damage of intestinal barrier, and then causes the colon cancer and then IBD things. And the way that we are trying to target is to look at healthy diets, which can improve the beneficial microbiota, or supplement with microbial metabolites, can that make it a healthy intestine, thereby increasing the healthy attenuation of tissue or organ toxicity. So the one of the microbial metabolite that we work on is called urolithin A, which is basically as a byproduct of eating the right berries or pomegranates, which are high polyphenolic rich diets. And you need to have specific microbiota or bacteria to convert or metabolize this allergic tannin or allergic acid diets into the so-called urolithin A. There are few bacteria recently identified which have not been tested at in vivo, but the study is still going on the actual responsible bacteria that is responsible for generation of urolithin A. So, I mean, in humans, if you take, I mean, the production of urolithin is not uniform, because take a 100 people, only for 35 to 50 people are only able to produce the urolithin, and also that also varies the different levels too. That potentially due to the differences in their gut microbiota can lead to differences in the urolithin production. But we still do not know how the levels of bacteria and then human population healthy versus disease have not been done yet. So what is urolithin A? It belongs to a class of organic compounds known as benzocoumarins or dibenzoalpha-pyrines. And ura is known to exert antioxidant, anti-carcinogenic, anti-inflammatory, and anti-aging properties. Some of the properties, such as anti-inflammatory, antioxidant, came from our lab, and it is bioavailable at micromolar concentration at colonic tissues without exerting any toxicity. We also synthesize the structural analog called USA3, is much more stable than the urolithin A, in collaboration with Dr. Vemula. And this is where I think I will start with a little bit of our published paper from our lab, where we showed that urolithin enhances the gut barrier function in IBD models. We did in TNBS model, this is the TNBS model, where you can see that TNBS causes the significant damage to the colon. Urea treatment or USA treatment kind of reverses the TNBS induced toxicity in this mouse model. It's not only in TNBS and also dextran sodium sulfate induced colitis, which is a chronic model which goes for 3 months. And I can appreciate where the DSS treated mice with the vehicle treated, you see a reduction in the colon lengths, a shortening of colons, and then urea user treatment reverse the effect of DSS induced colitis. The another activity that we look at is the gut barrier leakiness, permeability, where we can see that DSS induced vehicle induces a FITC dextran levels. Upon treat with the urea USA3, you see a significant decrease in the FITC dextran levels, suggesting that urea treatment definitely helped in reducing the gut permeability. And colon lengths and colon weight by lengths also improves upon treatment with urea USA3. And we basically we also worked out the mechanisms where we showed that uh, urolithin A definitely works through the receptor called aryl hydrocarbon receptor, and then activates the CYP1A1 and then goes through the NRF2 pathway to upregulate the tight junction proteins. We demonstrated this one by using the HR knockouts as well as CYP1A1 knockouts as well as the NRF2 knockouts, where the beneficial activities of urolithin A is mitigated in these knockouts. So overall, we proposed in that paper that urolithin A enhances barrier function through AHR, NRF2, as well as CYP1A1 dependent manner. So the hypothesis for this project started that urea USA treatment mitigates the arsenic-induced gut barrier dysfunction, inflammation, and toxicity in the gut epithelium. So first we wanted to establish whether arsenic causes damage, and then I mean this is done by Dr. Banerjee, my Banerjee. So we can see that these are the colon epithelial cells where we did the arsenic 24 hours. We can see that there are kind of arsenic-induced toxicity, but when we co-treated with urolithin or USA3, we see a protection from the arsenic-induced toxicity. I think you can much more appreciate at 48 hours and 72 hours. The cell viability increased. And then next we tested how it is doing. So arsenic is known to produce significant increase in the ROS production. So from control, we can clearly see that arsenic as the micromolar, you can see increase in the ROS production in a dose dependent manner. And upon incubation with the urolithin A, we see there is a reduction in the ROS production by and similarly, also arsenic induces apoptosis, and upon treating with the urolithin A, we see a reduction in the apoptosis. So suggesting that definitely urolithin A is affecting or protecting from RAS-produced apoptosis. And also we use the different method where we also look at DEFC, I think, the one of the thing that shows the if you increase the ROS, you see increased green fluorescence. But when we did the treatment with the urolithin A, we see that there's not much change in the green. That means the urolithin A able to block the arsenic-induced ROS production in this case. And the next to establish whether urolithin A has protects against arsenic-induced permeability or not, we use the assay called a permeability assays, where we use the plate the colon epithelial cells in the transwells and then grow for 14 days and then make sure that these cells form a nice monolayer, and then add arsenic with or without urolithin for 24 hours. If arsenic breaks the epithelial barrier, when we add the FITC dextran on the top, you see the FITC dextran in the bottom chamber. So when we perform the dose-dependent manner where we see that arsenic FITC with the dextran, you can see that as the dose goes up with the arsenic, the black bars, you can see the leakiness at the bottom chamber, suggesting that arsenic caused the damage in the barrier. And the next, when we co-treated with urolithin A, we can clearly see that there is a significant production of the Ph.D dextran in this one. The secondly, we also looked at urea dose dependent manner. You can see that urolithin A, when we kept the arsenic at 20 micromolar, and then we see that arsenic urolithin A at 25 micromolar, you see a nice decrease in the Ph.D dextran. And how it is acting, it's potentially, we said that one is maybe ROS-induced protection. And the secondly, also urolithin, as I mentioned previously, it has an ability to induce the tight junction proteins. The tight junction proteins are the proteins that really keeps the barrier more intact. Breakage in the tight junction protein causes the leakiness. And also, numerous reports suggest that these tight junction proteins are significantly lower in the IBD patients. So here, when we look at the control, you see a nice, this is one of the tight junction proteins which we looked for the protein called zonoclodine. You can see control, but when we added the arsenic, you see there is a significant decrease in the fluorescence. You can, and in addition of urolithin, there is an increase in the fluorescence. But interestingly, when we did even with arsenic plus urolithin A, we still keep the tight junction protein intact. So suggesting that, yeah, definitely urolithin A is mitigating the arsenic-induced tight junction protein disruption in this case. And the next, those are all the did it in the colon cancer cell lines, colon epithelial cell lines. The next, we move to look at what happens in the human 3D small intestinal organoids. So this is adapted from MatTek, where we culture the human small intestines in a 3D model organoids. They form all kinds of epithelial cell types, which like they can beautifully form villi structures, and they also have, you can see that villi green borders and called epithelial cells. You can see that nicely forms, and then they form a nice layers in the transverse. So in that, we tested whether urolithin A can have an impact and arsenic along with arsenic. So as the dose dependent manner, arsenic caused a damage in the epithelium and led to the Ph.D dextran leakage. And then addition of urolithin A in a dose dependent manner, there's a decrease in the Ph.D dextran leakage in these organoid structures, suggesting that yes, it is indeed urolithin A has a protective activities against arsenic-induced permeability. And also with the arsenic, you can also see that constant with the different doses of urolithin. Also, you can see that there is a protection against the arsenic-induced permeability. So that's basically, that was the paper that we published in archives and showed that at least in vivo and ex vivo model systems, arsenic can causes the damage in the gut epithelial barrier, and urolithin A has a protective activities against the arsenic-induced gut damage. The next question we asked is, okay, how does it really work in vivo model systems? For this, we have to shift to a little bit different model system, because a regular C57 black 6 do not, they basically clears up the arsenic much quicker than the humans, because arsenic, the enzymes responsible for the methylation of arsenic for the excretion, that's called AS3MT, acts much better way in mice compared to the humans. So you really don't see in C57 black 6 effective arsenic related toxic effects. So that really hampered significantly the arsenic studies, preclinical models to really examine the mechanisms. So as I mentioned, methylation of the arsenic by AST3MT controls arsenic detoxification and modifies risk of arsenic-induced diseases. So arsenic comes from different forms where it forms the monomethyl or dimethyl or dimethyl arsenic, and it's all mediated by the AST3MT, both at the oxidative methylation or reductive methylation. So as I mentioned previously, the knockouts of AST3MT knockouts are definitely more susceptible to the arsenic-induced toxicity in mice compared to the uh, regular wild-type mice. So as I mentioned, that if you look at this lack of AST3MT knockout mice, you can clearly see that knockouts in the urine, you see that they are excreted very less compared to the wild type mice, both males and females. But in the livers, you can see that there is a significant accumulation of arsenic in these knockouts. So the arsenic AST3MT knockout mice have been good models to study the accumulation of arsenic in the systems. But it is a knockout, so really you really can't really say that what's really happening in the humans. So to overcome this, Dr. Stiblo's group from UNC, they created a novel mouse, which is they replaced the human, they replaced the mouse AST3MT mouse with the human AST3MT gene into the chromosome. So basically in the humans, as I mentioned, arsenic, when you give it to the both humans or the mice, you really don't see inorganic arsenic or methyl organ or um, accumulated in the mice. They do not accumulate much, but whereas in the humans they do. So to overcome that, they took out the mouse AST3MT, replaced with the human one, and now these mice can now accumulate the similar levels of inorganic arsenic and the methylated forms of arsenic in mice. So basically, you are recapitulating the are mimicking the human arsenic metabolism in the mouse. So these mice are generated last year, I think last year. I think as soon as it kind of Dr. Stibler is kind enough to give us those mice to us, so we bred and we maintained the mice at our facility. So the little bit by characterization of this humanized AHA mice, there is a significant difference between the human mice in efficiency, arsenic methylation, and this is the paper they published. And, and in that, they characterized where they can clearly see that these are HSM means the humanized AST3MT mice, and these are the wild type mice. So in the urine, you can see that the wild type mice are clearly excreted much better than the AST3MT, humanized AST3MT mice. And the liver, you can see that they definitely is accumulated more in the liver. So this basically is mimicking the more or less human arsenic levels. So mentioned we kind of showed that urolithina can attenuate the arsenic-induced gut barrier dysfunction. So as I mentioned, the data that we showed that arsenic can cause the oxidative stress, which potentially causing loss generation of apoptosis, loss of TJ protein and inflammation markers, increased inflammation when we did the urolithin A, and we reduced oxidative stress and protected from or blocked all these activities. Now we wanted to see whether is this true in the in vivo. So first thing that we wanted to uh, check is whether arsenic induces tight junction proteins in the mice or not in this humanized AST3MT mice. So this is very low level of arsenic been exposed to these ones. We got the intestines from Dr. Stablo, and then these are 20 weeks exposed, and we can clearly see that tight junction proteins called zonocludin, occludin, or claudin 4. You see there is a reduction in this tight junction proteins. No, I mean that's normal. When we exposed to the arsenic, you see a uh, degradation of this or reduced levels of zonocludin and the tight junction proteins compared to the uh, non-exposed arsenic. So there is a gut barrier damage is happening. So we got those mice. It took a long time to generate these mice. Okay, it took almost a year to get the number of mice that we required. So finally, we performed whether looked for the almost 10 weeks. We exposed these mice with 400 ppb, and and these are pure diets, so that there's no arsenic contamination in those diets. And then we treated with either vehicle or urolithin A 3 times a week, and then we euthanize the mice. We can see that these are control colons and just a vehicle and urolithin. But when we expose the arsenic, we can definitely see there is a shortening of colons and compared to the control groups. And when the mice that the cohort that is treated with urolithin A, at least protected from the arsenic induced shortening of colons. Can clearly see that here colon length measurements and colon weights by length. Weight really tells us how much inflammation is there, how many inflammatory cells or any kind of things are happening. And we can clearly see arsenic definitely increased the colon weight by length, and urolithin treatment mitigated that function. And the standard assess we do is the permeability. We can see there is a definitely, it's not much, but there is a still compared to the controls, there is a leakiness in the gut where we see increased fitzy-dextran in the serum. And whereas in the mice that are treated with urolithin A has the significantly lower than that of arsenic exposed to mice, suggesting that yes, there is a uh, gut leakiness happens and the urolithin A treatment protects that. The next, we also checked the inflammatory markers, whether arsenic is not induced inflammation. So in the serum, when we look, not in, these are all the colons, you can see that there is an increased IL-6 at the colon sites, and then urolithin definitely reduces, and TNF alpha causes, arsenic causes increase in TNF alpha, this reduces, and IL-1 beta. And MPO is a myeloperoxidase assay where one can measure the recruitment of neutrophils at the colonic sites. So arsenic exposed mice showed increased MPO levels, and then treatment with urea definitely reduced the arsenic. Right now, we are in the process of characterizing the sections, H&D, and then staining the sections to see where and how much damage that is happening. And it's the next thing also, we looked at the tight junction proteins in these mice, and then we see that the zonoclodin or oclodin have reduced when the mice are exposed to the arsenic, and then urolithin at least recapitulate, at least restore the expression of tight junction proteins here. This is the estimation of Western blots where we see that at least decreased and at least restoration of the tight junction proteins. We also looked at the RT-PCR where we see an increased, decreased occludins, and the urolithin causes the restores the occludin and zone occludins. So then I mentioned the neutrophils play a role. I mean, then we looked at flow cytometry by looking at PMNs at the spleen as well as mesenteric lymph nodes. We see there is an increase in the neutrophilic population, and the overall inflammation is gone up. And also at the mesenteric lymph nodes also we see an increased neutrophils, and then urolithin can cause the reduction in the neutrophil. But how exactly it is doing, still we are trying to figure it out. That is grant whether the neutrophils have a role in GI related functions induced by the arsenic. And we also did microbiota sequencing. Still at the bioinformatics group to really further analyze, but the basic characterization is very interesting to see that the the genus really kind of Rhombostritia is completely eliminated in arsenic treated mice, but when compared to the arsenic, when treated with urolithin A, kind of restored, similar to the control. I really don't know what this bacteria is doing there. I'm still kind of figuring it out. And then Akkermansia also is kind of, I mean, the role of Akkermansia is always a questionable to me. It has the potential beneficial activities. At the same time, it can also cause damage. So, but it's not that significant, but definitely this is something which really attracted my eye, which we really wanted to do further analysis and further see if we can get this bacteria and then see if it is this responsible for this damage or not. Now, we also gave this, again, thanks to CIEHS pilot program, and we also gave this for RNA sequencing for these samples, the colonic samples, to see really what really happens, how this arsenic is causing the GI-related damage, and then how urolithia is able to bring back. If you get some clues, that would be our aim to go for a grant. The second thing, we did this human organoid cultures, as I mentioned, we did our RNA-seq. The data is too good to believe, so just had to really analyze. I think why you did this analysis. I mean, the pathways I know there are a lot of pathways that are there, and I'm really confused whether I can really pull through what they are. There is a significant compared to control. Arsenic changed so many things, and when we did the urolithia, this whole thing is disappeared, and then then other things came up. So right now, I'm still looking at the clues where we had to focus on this aspect. So, I mean, I just based on this, not all of it, based on this data, the kind of proposal or the kind of things that we uh, speculated or hypothesized is that how urolithia is really kind of mitigating the arsenic-induced toxicity in the intestinal cells. So is it really working on at the, of course, epithelium level, or is it also have an impact on the microbiota level, where is it affecting on the arsenic uh, metabolism or not? So for this purpose, we proposed in the recent grant where the UROA has a role in uh, in the arsenic speciation or not. So for this, we kind of culture the human microbiota in a mini bioreactor systems, presence or absence of arsenic and urolithin and identify if any uh, changes that we can observe with arsenic speciation. And the second thing is similarly, whether arsenic induced causes the damage, and we will use a human intestinal organoid system to see if any, again, arsenic species changes or the drug transporters, if it has an impact on that. And the second one is a little bit more ambitious, but if the funds are available, this is something which really I wanted to test is we have a humanized AST3MT mice here, but the microbiota is still from the mouse. So, I mean, in the humans, we do know that microbiota has a role and they have a different types of microbes. So, and we wanted to generate a humanized microbiota mice in the context of humanized AST3MT mice, and now test how the arsenic exposure affects the GI function, arsenic metabolism, and how does urea treatment would help in reducing the arsenic-induced toxicity. So based on that, we thought we will get a clear idea, at least some, how this arsenic is really damaging the GI functions, and the utilization of these beneficial metabolites would have a better effects. So that is the project that thus far I did with funds that are available from CIEHS. Thank you once again. Without that center, I wouldn't be able to do all this stuff. So the take-home message is arsenic-induced gut barrier dysfunction, and it disrupted the tight junction proteins, microbial metabolite, urolithia protects against arsenic-induced toxicity. and humanized my AST3MT mouse column. So established at UFL and tested for the protective activities of urea. Good thing about this mouse model system, the day of euthanization, we gave all the tissues to different PAs in the university, so they can at our center, so that they can look at how this arsenic is affecting the other organ functions. The future directions was identify molecular cellular mechanisms and to look at urea and arsenic metabolism, define the role of human microbiota in arsenic metabolism. It's that's one of our projects. We also look at mechanisms of IBD and alcoholic liver disease and colon cancer too. How this? I know. I mean, when I talk about urolithin, it can do so many things, and what really it is doing, I still don't know because the literature is exploding like anything. Just as an immunity paper showing that it has an impact on the CD8 anti activating the CD8s to work against the anti-tumor activity. So yeah, in the review, it's kind of shows, I mean, it has effect on the whole organism where increases the longevity, muscle function, heart, brain, joints, immune system, intestines, and liver, pancreas. We published the pancreas hepatitis recently also, and kidney function. We try to see that we did the experiments, but no personnel to really take the other level. So we are focusing on these two things. And joint cartilage, we are collaborating with a few others. Let us see how that goes. Yeah. Yeah, I mean, ideas are there, but it's just other things. So yeah, it has significant impact. So the major acknowledgements, definitely this project started with CIEHS, and then Mayuk and Dr. States definitely helped me to move forward this project. And all the work is done by Dr. Ghosh. Without her, this project never existed. And Jack worked with animals and helped us to take this project to the next level. Funding, definitely, I've been telling CHS, that's the only one thing I want to do this work. So yeah, Cobra helped me to at least to maintain the mice for some time. So that's definitely so. Happy to take the questions, and then I'll stop it here.